Good day, fellow scientists and other curious minds. In today's video, I'll be demonstrating some techniques used in the field of wastewater-based epidemiology. Wastewater surveillance has been around for almost a century, back when it was first used to detect polio. More recently, due to the COVID-19 pandemic, these techniques have become modernized, using sequencing and qPCR to both identify and quantify the amount of viral RNA in wastewater all over the world. One of the biggest findings to come from the Ottawa Wastewater Initiative was that SARS-CoV-2 RNA is detectable several days before hospitalizations begin to rise. This sort of data is used to provide health information to the general public and healthcare facilities so that individuals can stay aware and hospitals prepared for increased burden. Now that the pandemic is over, wastewater surveillance is still being used to track seasonal diseases like flu and RSV, and is also being used to understand the agricultural impact of the recent avian bird flu. But for now, let's head over to the bench and take a look at the experiments that produce this sort of data in more detail. Let's first introduce the types of samples we'll be working with. It all starts with infected individuals who shed viral particles that end up going down the drain. This could be from their own home, hospitals, schools, or airports. As these particles travel through the sewer system, their DNA or RNA is somehow protected, and eventually, this virus-containing wastewater ends up at the treatment plant. This is a bird's-eye view of RT-qPCR. It is the main wastewater treatment facility in Ottawa. Two types of wastewater are routinely measured for viral RNA. Influent, which is water that flows into the facility, and sludge, which is taken from the primary clarifier within the plant. Here is an overview of the workflow that will be showcased in this video. First, RNA is extracted from the wastewater sample. Next, the extracted RNA is purified to remove contaminants and inhibitors. Once the RNA is purified, we can quantify the amount of viral RNA in our sample. We will go over how to extract RNA from two types of wastewater, namely sludge and influent. Then demonstrate the purification process that uses silica columns for both of these sample types, and measure the amount of disease-specific RNA through RT-qPCR. Let's start by first taking a look at how viral RNA is extracted from the solids collected from primary sludge. We begin this protocol with a sludge sample. The solids are collected into a pellet by centrifugation, and the bulk of the water content is removed. Next, we add glass beads and buffers from a QIAGEN kit to help with extraction. The sample is then homogenized by bead beating to release RNA from the sludge solids. This is a sludge sample. It is a semi-solid material that accumulates within the primary clarifier of the treatment plant. Sludge is too thick to pipette normally, so you have to cut the pipette tip. It is best to use a filter tip here in order not to contaminate the pipette. Approximately 1 milliliter of sludge is added to a tube. The tube is then centrifuged at 15,000 times gravity for 10 minutes to collect the solids into a pellet. After discarding the supernatant, the sample is ready for homogenization or for storage at -20 degrees. First, quick spin the glass beads to collect them in the bottom of the tube. Next, add Trizol to the tube. This will help protect extracted RNA by degrading nucleases. Transfer the sample pellet to the tube with the beads and Trizol. Finally, add freshly prepared buffer to the tube and proceed directly to homogenization. Homogenization is achieved by chemical and mechanical forces, as you can see here. The tube is shaken vigorously, causing the beads to fly around and smash apart the solid sample, while RNA is extracted into the buffer. The sample should appear as one single homogeneous phase after bead beating. Centrifuge the tube again to separate the buffer and the destroyed solids. Note that after centrifugation, the supernatant is still not very clear at this point. Transfer it to a new tube. The supernatant gets mixed with an inhibitor removal solution that causes debris and contaminants to precipitate out. Mix this solution thoroughly by vortex, then incubate at 4 degrees for 5 minutes. Another round of centrifugation will pellet the contaminants and insoluble material. Transfer the clear supernatant to a new tube. Add buffers and ethanol, then mix by vortex. The sample is now ready to be loaded and purified on the column. Now that we have seen how RNA is extracted from sludge solids, let's go over how to extract RNA from a liquid sample, such as Influent. This protocol begins with an Influent sample. An aliquot of the sample is taken, and series magnet beads are added. Viral particles become concentrated on the beads through affinity capture. Wastewater is removed from the sample, and particles containing RNA are eluted off the beads. This is an Influent sample. It is a liquid sample with trace amounts of solid material and is taken from the main inflow of the wastewater treatment plant. Start by transferring some Influent to a new tube. This can be scaled up or down accordingly. Series bead technology is used to both concentrate and extract viral RNA from the sample. First, add enhancement reagent to the sample, which improves the RNA binding to the magnet beads. Vortex to mix. Next, add the magnet beads. These appear as a brownish-orange slurry. Invert several times to mix, then incubate the sample at room temperature for 10 minutes. Invert the tubes to mix again at the 5-minute mark. After the incubation, place tubes on a magnet rack to separate the now RNA-bound beads from the liquid sample. It usually takes 1 to 3 minutes for the magnet beads to fully separate. The solution should become clear again, and the beads should accumulate up the sides of the tube. Remove the liquid sample, being careful not to disturb any beads from the walls of the tube. Remove the beads from the magnet, resuspend them using pure water, and transfer to a smaller tube. Separate the magnet beads again using a smaller magnet rack. The beads will now stay bound to the back of the tube while on the rack. Remove the water. Resuspend the beads using a small amount of PBS mixed with lysis buffer. And incubate for 10 minutes at room temperature to elute the RNA off the beads. Bind the beads to the magnet rack again and transfer the supernatant containing RNA into a new tube. Finally, add ethanol to the sample and mix by vortex. The sample is now ready to be loaded and purified on the column. Regardless of which sample type the RNA has been extracted from, it still needs to be purified before analysis. Let's take a look at how this is done with silica columns. This protocol starts by passing our extracted RNA sample through the column. RNA stays bound to the column and wash buffer is passed through it. After the RNA is washed, it is eluted off the column with pure water. First, spin the sample through the column. All the liquid should transfer through the top chamber into the bottom of the tube. The RNA is now bound to the white silica part of the column, while the ethanol and other buffers pass through. Discard the flow-through and then add wash buffer to the top chamber of the column. Centrifuge the tube again to pass buffer through the column. Discard the flow-through and centrifuge to dry the column of residual wash buffer. Since we are interested in isolating only RNA, we add diluted DNAs to the column to degrade DNA. This occurs during a 15-minute incubation at room temperature. After this incubation, wash the column again to remove the DNA's solution. Discard the flow-through. Spin the tube again to dry the column. Transfer the column to a new collection tube Then add pure water or tris buffer directly to the column. Be careful not to poke the column with the pipette tip. Let the column incubate for 1 to 2 minutes at room temperature, then centrifuge the tubes to elute the RNA. Carefully orient the cap of the collection tube in the centrifuge so that it does not break off during the spin. Discard the column. This is now your purified RNA sample. Check the yield and purity of each sample using a nanodrop. Several hundred nanograms of RNA are expected, along with a 260-280 ratio of 2.0. Our RNA has now been extracted from our sample and purified. We are now ready to quantify the amount of viral RNA extracted from the sample using RT-qPCR. A reaction mix is prepared that contains everything needed for RT-qPCR. The extracted RNA samples are then added to the reaction in 96-well plate format. Samples are then run on the thermocycler and RNA is quantified. We prepare this mix using pure water, primers, and probe that are specific for our disease target, our RNA sample, and a special buffer that contains everything needed for reverse transcription and PCR. Combine all of these reagents into a fresh tube, mix by vortex, and quickly spin to collect all of the volume. Load 7 microliters of the master mix into each well of a 96-well plate, followed by 3 microliters of RNA sample to each corresponding well. Lightly tap the plate on the bench to collect droplets at the bottom and seal the plate. Give the sealed plate a quick spin to ensure all the reaction volume is mixed in the bottom of each well. Load the plate into the thermocycler with the proper protocol and wait for the plate to be read. This is what the typical data curves look like for a qPCR assay. The main readout of a qPCR assay is the CQ value, which stands for cycle of quantification. This is the cycle number where the RNA signal passes the threshold of becoming detectable. CQ values are counterintuitive because a lower CQ value actually means you have more RNA. The higher the CQ value, the less RNA is present in the sample because it will take more cycles to amplify and become quantifiable. For example, the first point in my standard curve is around 600 copies per microliter and gives a CQ of 29. The last point of the standard curve is 7 copies per microliter and gives a CQ of 36. Using this information from the standard curve, we can extrapolate a value for our unknown sample. If our sample gives a CQ of 33, we can calculate that it has around 55 copies of RNA per microliter. Once the amount of viral RNA in a wastewater sample has been quantified, the data is submitted to wastewater surveillance networks. On a provincial or city level, individual institutions can provide high-resolution data, usually daily or weekly, to produce and track trends over time, as seen in this example from the University of Ottawa. You can clearly see the rise and fall of peaks throughout the pandemic, which correspond to different outbreaks and variants of concern. These types of data are conglomerated by the Government of Canada and are summarized in a dashboard that gives a countrywide view of the present and past levels of SARS-CoV-2, influenza, and RSV. If you live in the US or the EU, similar dashboards can be found through either the CDC or the European Commission. I hope this video has given you a better idea of what goes on behind the scenes in the labs working on wastewater surveillance. Check out your region's dashboard the next time flu season hits and see if it aligns with your perception of the general public's health status. Thanks for watching, and good luck with your experiments.